Hi, this is Dr. Rudresh. Welcome to my channel Medical Microbiology Guide. Today I will be discussing on Widal test. Widal test was first introduced by Georges Fernand Isidore Widal. So the test is based on the principle of tube agglutination. So there are various tests which are available based on the same principle. They are called as febrile agglutinin tests. So they are done for detection of the Salmonella, Rickettsia, Brucella and Francisella infections. So in Widal test we are going to detect the antibodies against the O and H antigens of the typhoid and paratyphoid bacilli. Usually the test is recommended to be done in the second week of illness when the antibody titers are high, and the sensitivity of the test varies from 46 to 94%. So let's briefly look at the structure of the Salmonella. Salmonella is having two types of antigens, O antigen, which is also called as somatic antigen, and H antigen, which is known as the flagellar antigen. The O antigen, it occurs on the surface and it is made up of the specific sugar sequences. O antigen is usually less immunogenic and it's a lipopolysaccharide complex. It is heat stable and alcohol stable, but and it's also resistant to 0.2% formaldehyde. Okay. The antibodies against the O antigen usually be detected after 6 to 8 days of onset of the fever. For the Widal test, the O antigens will be stained with a blue dye. The H is a flagellar antigen. This is actually genus specific. It is heat and alcohol labile. Usually the H antigens are preserved in 0.2 to 0.4% of formaldehyde. They are strongly immunogenic and the antibody response usually appear after 10 to 12 days of the onset of the fever. There is one more antigen which is known as VI antigen. The VI was named because it was earlier assumed to be associated with the virulence. This usually overlie on the O antigen and it is present in few serotypes like Salmonella typhi, paratyphi C and Dublin. The VI antigen, it actually renders the bacteria inagglutable by their specific O antiserum. So, and also it's poorly immunogenic. If you find the antibodies against the VI antigen even after the early convalescence, it indicates that the person is going for development of the carrier state. So in Widal test, we are going to employ only one O antigen and we are going to detect the H antigens of the typhi, paratyphi A and B. So we are using only one O antigen because the factor 12 of the O antigen is shared by all the three species of the typhoid, that is typhi, paratyphi A and B. Hence, because of the common factor 12, so we use only one O antigen and different H antigens based on the different species. O antigens are usually prepared by culture of the Salmonella typhi on phenol agar, and H antigens are usually prepared by adding 0.1% of the formalin to 24 hours growth culture of the Salmonella of respective species. Let's see the procedure. So we require the test serum which should be fresh. Usually if there is a delay in the test, we can preserve it at 2 to 8 degrees Celsius for up to 72 hours. So generally hemolyzed samples are not suitable for the test. The antigens, that is commercially available or the in-house prepared ones, which the O, H, AH and BH antigens. So we need normal saline for the dilution of the serum, water bath for the incubation, Pasteur pipette with the bulb to for the dilution, doing the dilution, and test tube rack. And we use two types of the tubes, that is Dreyer's tube and Felix tube. The Dreyer's tube is used for the H agglutinin, which is a narrow tube with a conical bottom, and the Felix tube is actually short and round bottom tube. So there are various commercially available antigen kits are available, so we can use any of those kits. So initially, so for doing the Widal test, we are going to take 0.9 ml of saline in the first tube, and 0.5 ml of normal saline in the rest of the tubes. So here I am showing dilutions starting from 1 in 20 to 1 in 160. So if you want your final dilution to be 1 in 20, so we have to prepare an initial dilution of 1 in 10 in the first tube. So we are going to add 0.9 ml of the saline and 0.1 ml of the serum. So totally it become 1 ml. So 1 part of the serum and the 9 part of the saline, making 1 in 10 dilution of the serum. So now we are going to transfer 0.5 ml from the first tube to the next tube, which contains 0.5 ml of the saline. So when you mix it, it becomes 1 ml of the 1 in 20 dilution. So the doubling dilutions of the serum is done with all the tubes, and the last 0.5 ml will be preserved if it is required to do the extended Widal test. So once we have made the dilution, so we will be left out with 0.5 ml of the each dilution starting from 1 in 10 to 1 in 320. Now we are going to add 0.5 ml of the diluted antigen to each of the tubes. So when you add 0.5 ml to the existing 0.5 ml of the diluted serum, so it becomes total 1 ml, and the further double dilution of the initial dilution will occur, giving rise to a final dilution of 1 in 20, 1 in 40, 80, 160, 320 and 640. Now we are going to incubate in the water bath in such a level that the water level should be half of the fluid level in the test tube. So this will create a water current which will make the antigen antibody reactions in more efficient, and they will settle down properly at the bottom. So we are going to observe for the agglutination and the clumps of the antigen antibody clumps in the bottom of the tube. So generally it is preferred to see with the mirror so the highest dilution of the serum which gives the agglutination is known as the titer. So you can see, so it starts from 1 in 20 to 1 in 640. So at 1 in 160 the agglutination has stopped, indicating that this is a titer for the particular serum. So this is a Widal rack with the tubes arranged. So with each test we are going to put control, okay, control for O and control for H antigens, which is nothing but a saline control. So the O antigen will give a compact granular agglutination, whereas H antigen will give loose cotton wool clumps. So this is a test serum which shows an agglutination till 1 in 320. So we can see the clearing of the solution as well as the sedimentation of the antigen antibody complexes to the bottom. So this is a saline control which shows the turbidity and no only a small button formation. So by looking into the clearance of the solution as well as the sedimentation of the antigen antibody complexes, both will indicate the presence of agglutination. So we are going to see till what tube the agglutination was seen. Okay, if we are seeing till the end, we can go for the further testing of this the diluted serum which we kept in the during the procedure. So this is a negative test for O antigen. You can see there is no clearing as well as only a small button formation is there, both with the control as well as the test. So this is the H antigen. So we are seeing the agglutination is also seen here till 320. So we are seeing the clearing as well as the sedimentation of the antigen antibody complexes. Again, this is a negative test for the H antigen. So the one thing, one precaution should be taken is you should not shake the tubes before the reading of the results are done. So control tubes should show no agglutination, and the test serum dilution, we are going to look for the highest dilution of the serum which is showing the agglutination, which is nothing but the titers of that particular serum. Usually the we know the antibodies usually appear after the first week, and they increases for third to fifth week, then they start declining. So hence it is ideal to demonstrate in the second week, and we are going to take one more sample after 7 to 10 days. Okay, so if you are demonstrating a fourfold or more rise in the titers, so then we are going to say that the person was infected. This is because if you do a single test, the baseline titers for each region it varies. Again, the different places of a same metropolitan city may show different titers because of the variation in the occurrence of salmonella in that particular population. Hence, the baseline titers for a particular region should be known before the interpretation of the Widal test. Generally, we take at the antibody titers of more than 1 in 80 and H antibody titers of more than 1 in 160 are suggestive of the enteric fever. So if you do a paired serum samples, that is one in the second week, one after the 7 to 10 days of the first sample. So if you demonstrate a fourfold rise, so it indicates that the infection is going on currently. Some textbooks they mention only 2 to 3-fold rise in the titers can also be taken as a positive Widal test. So there can be a false positive Widal test in case of the non-typhoidal salmonella infection, and also in case of chronic salmonellosis associated with the cystosomial infection. So in case of vaccination with the TAV vaccine or typhoid vaccine, in some infections like typhus fevers, acute falciparum malaria, dengue, chronic liver disease, rheumatoid arthritis, the myelomas, okay, and nephrotic syndrome, so there we can see false positive results. False negative viral results can also occur if you treat the case in the early. Okay, so the early treatment will prevent the much of the immunological response, thereby decreasing the titers, and it will be shown as false negative results. Then following a typhoid relapse, so relapse will weaken the immunity, thereby decreasing the amount of antibody produced in the body. And severe hypoproteinemia, carrier status, and sometimes technical errors can give rise to the false negative Widal results. So that's going to finish the Widal test. So thank you for watching the video. Please subscribe my channel, like this video, and press the bell button for the future notification of my next uploads. Thank you.